So, Kathleen, would you like to take it away?
Yeah. All right. It's nice to be here today and talk to you all about this technology that I started using in my second year or first year of my PhD in Eleanor's lab for our Darwin's Ark project, studying domestic dogs, pet dogs that we survey from owners. And we needed a strategy to be able to get genetic information from thousands of pet dogs. Some genotyping chips, which test for specific sites on DNA, exist for dogs, but we didn't really think that would be sufficient for discovering new genetic associations. And a lot of those arrays were built with the breed populations in mind. And we had a lot of mixed breed dogs and dogs of diverse ancestries involved. So we switched from dense genotyping to low coverage sequencing as a strategy for genotyping thousands of dogs. And it's been working out so far, so good. I wanted to tell you a little bit today about why we chose that and kind of what the differences are between doing high coverage, low coverage, or genotyping arrays. So in an ideal world, we would, for our population studies, we would get the highest confidence genotypes for all of the variation across the entire genome for all the individuals that we ever want to study. But in reality, budget limitations lead us to having to kind of pick the best 2 of 3 options using different technologies. The high coverage genome sequencing approach, whole genome sequencing, will of course get you high confidence genotypes for a large fraction of the genome, but it's prohibitively expensive to do this for more than a handful of individuals. So when we're looking at population studies in the past, most people would choose genotyping arrays, which are specific products chosen to assay specific sites on the DNA for a given species. These will get you very high confidence genotypes for those specific sites. You can do these for a large number of individuals because genotyping arrays are fairly affordable, but they come with their own population biases and limit you to those sites that the genotyping array is designed for. Low coverage sequencing, on the other hand, is whole genome sequencing, but taking a lower pass at the genome, getting 1 to 2 or fewer, sometimes no reads, at all of the genome rather than specific sites. And the benefit of this is that you're getting more information covering a region than you are with genotyping arrays. But what you sacrifice here is possibly the high confidence genotypes at every single site. There is the possibility that genotypes in a not well covered region for between individuals will have lower confidence genotypes as a result of this method. And not all the individuals will have the same level of confidence at all the genotype sites. That's okay for a population study. I think in human applications and applications where you want to return information to individuals that's more confident, people opt towards genotyping arrays because you get that high confidence at the sites you care about. But for discovery and population studies, this is really good enough. And I'll talk about a little bit of why that is. So some of the features, advantages, and trade-offs of low coverage sequencing. The first feature is it uses short read sequencing technology. So this isn't a specific product that a company makes for you. You don't have to rely on a company to develop and sell an array product. If you have a species that doesn't already have an array, if you wanted a genotyping array, you'd have to work with a company to develop a product. And then you'd be stuck purchasing that product from that company. Sequencing doesn't have that limitation so much as you're limited to, say, Illumina sequencers. So you can shop around a bit more. The trade-off is that if you're going to do this completely independently, you have to have some sequencing experience. That said, there are companies like GenCove Incorporated, which developed the imputation method that will do kind of this process for you. You can work with companies to do the sequencing prep, and the imputation. But if you want to do it independently, which you absolutely can, you might need some sequencing experience. In low coverage sequencing, genetic variants are what we call unascertained. You're not predetermining the variants you're looking to discover in your population. You're not saying this is the set of single nucleotide polymorphisms I want to study in the species. They're not determined ahead of time. You have a reference panel that you develop and discover variants from and might use to aid in imputation of low coverage data. But the quality of your data is not tied to some population bias that you predetermine by selecting genetic variants. But that said, the limitation of low coverage sequencing is that not all the samples will have the same sites competently genotyped. So there's going to be variation in how well you're called for each individual. Your low coverage data is going to improve as your high coverage reference data improves. So there's kind of this future proofing of your low coverage data. Earlier arrays, less dense arrays, are always going to be limited by the number of sites they happen to genotype at the beginning. High density arrays were selected to improve that, but they're still limited to those markers. Your sequencing data can always be reimputed and reevaluated in the context of new data. But that also means that your data quality is somewhat tied to your reference panel size at the time of analysis. And there is no sequence specificity for low coverage sequencing. So the microbiome, say you're taking saliva samples from an individual or skin samples, can be captured in some cases. So that could be a potential bonus if that's something you're interested in for your species. It could be also a potential source of sequencing failure if the microbiome is very prevalent in the sample you're taking. And that is a source of sequencing failure by outcompetition of the microbiome reads. The pathogens or microbiota are outcompeting DNA from the individual or host you're interested in. So there's a balance there. So I wanted to talk a little bit about how imputation of genotypes works from low coverage sequencing. So low coverage sequencing on its own produces raw sequencing reads, short sequences from DNA, but to actually get the genotype calls, we use a statistical method called imputation. So what you have in most cases is a reference panel of genomes you've sequenced at higher coverage, maybe over 30x, or 20 or 30x. And from this, you've called variants, genetic variants, and you've determined phased haplotypes, basically order of sequences that are present in your reference samples. In this case, I'm marking the non-reference alleles with black, the reference alleles with gray. And this is kind of a small example because sequencing reads are much longer than this. But when you're taking samples from a diploid individual and either going the path of low coverage sequencing or array genotyping, with sequencing reads, you're getting reads that overlap with the region of interest. And you are inferring which haplotypes the individual likely has based on observations in those reads. So you have a little bit more information to work with there. Sometimes you'll get 2 times coverage. Sometimes you'll get 1 times coverage. Sometimes you will get only 1x coverage or no coverage, in which case it's equivalent to kind of having untyped variants there, and your imputation might suffer as a result in particular regions. But you are capturing the whole genome, so this isn't the case everywhere. In array genotypes, you are necessarily dealing with untyped variants, and your imputation is based on the typed variants, which will always be somewhat limiting. It's kind of like a dearth of information in those untyped regions, whereas at least with low coverage sequencing, you're getting the opportunity to have more information to support your inferred genotypes. At a larger scale, a lot of these genotype imputation methods also consider recombination as an effect. So a lot of these are based on hidden Markov models, something that's basically tracking a sequence. And there is an emission probability or the chance that this haplotype is going to switch, and it's built into the model to detect when haplotypes change. And certain methods will employ recombination hotspots, or will just consider a constant rate of recombination in the genome. Now I'm going to talk about 3 different methods. And the first I thought might be very relevant for species that are underrepresented in sequencing data, and that's the method called STITCH. The strength is that this method requires no reference panel. That high coverage reference panel I mentioned earlier, you don't need it to run STITCH. This is good news for non-human and non-model species that don't have that high coverage data available. It relies on the hidden Markov model. So it's basically studying your query set of data, your low coverage sequencing, data and trying to discover what the ancestral haplotypes are given a number of presumed ancestral founders. And it just iterates over the query data to discover these haplotype states. So I kind of like that approach. You're learning from your own data. It's computationally heavy. The time it takes to impute genomes using this method scales linearly with the samples and the number of variants, and quadratically with the number of ancestral founders. And it's really limited to common variants. You're not going to be able to support rare variants without high coverage reference genomes. Another method that came out is the commercial GenCove low impute, which has a strength of directly inferring genotypes from the mapped sequencing reads. So you don't have to do any variant calling beforehand. It's also a hidden Markov model, and it employs this haplotype copying model very similar to what I was describing from a computational method by Lee and Stevens et al. It's computationally heavy as well. It has some workarounds to reduce the computation time, such as downsampling the number of haplotypes in a given region based on the query data. And it's great for common variants, and it's also pretty good for rare variants because it'll prioritize the reference haplotypes that share rare variants with the query set of reads that overlap the region. The newest method to imputing low coverage sequencing data is GLIMPS, which stands for something, but it didn't actually work as an acronym. So I don't know, but the strength here is that they actually sought out to improve upon all the existing low coverage sequencing techniques or low coverage imputation techniques. So it improves upon all the current methods. It can also handle haploid, diploid, or mixed ploidy species without doing any splitting of the data. It's also a hidden Markov model. It's a good approach to imputation. It's a similar modeling approach to low impute, same principle, except the inputs are genotype likelihoods, which means you do have to do variant calling of your data before you put it into GLIMPS. It also adds steps that improve the phasing, which is the separation of the maternal from the paternal haplotypes. Computationally, it sought out to improve the inefficiencies of other methods, so it's actually very fast. What they say is that it costs less than $1 to computationally impute your data using GLIMPS, and it's extremely well benchmarked, and they did a great comparison for human data, low coverage sequencing with imputation with GLIMPS versus all of the genotyping arrays that have ever existed for humans, and their performance on European samples, African samples, Asian samples, and showing how low coverage sequencing can remove that population bias of array genotyping. And it showed that they did outperform other methods for rare variant detection. I would also note that low impute, you do have to get, I think, approval to use. It's free to use in public, but you have to request it. GLIMPS, I think, is publicly available. Stitch is publicly available, but it's from 2016, and there's a tendency in bioinformatics world to abandon software, so I don't know how well supported it is currently. What I really liked about Stitch's approach is that it was learning from the query data. So in some studies, as you accumulate more and more low coverage sequencing data, like we have in our dog project, where we're at almost 3,000 samples, we'd really like to know if we can learn anything from the data we've collected, rather than always looking just back at the high coverage sequencing data. So that's one thing I don't think imputation methods have employed yet, is how to learn also from the query data. From GLIMPS's paper, they did a comparison of the performance at different genetic variant allele frequencies, so rare variants versus common variants. Sadly, Stitch does not do very well, except for very common variants. And GLIMPS tends to outperform most of the other imputation methods. The running time, of course, also, they cut this down incredibly, so it's very computationally efficient. And I wanted to close off by talking about how low coverage sequencing is already being used to study climate-related population crises. So this is not my work, but recently published work studying bleaching of coral. There was a bleaching event in 2017 in the Great Barrier Reef, And scientists went out and selected about, I think, 4 branch fragments from 12 different sites along the Great Barrier Reef and phenotyped the degree of bleaching visually and through other metrics at these different sites. This is the population genetics of coral Acropora millipora toward genomic prediction of bleaching. And there's no getting around this. You do need a reference genome to do any sort of sequencing study in your species. That's going to be a big upfront cost as they assembled a de novo biogenome. They did do a reference panel. So they sequenced 44 of these samples at over 20x coverage, which is going to run up costs quite a bit. But subsequently, they can sequence the rest of the samples at 1.5x coverage for much lower cost. And they made sure that their reference panel represented all 12 different reef sites. So you're getting the diversity of population structure there. And while they didn't find any common major effect loci associated with the variation in bleaching, they did find a few genes that seem to be associated not significantly with bleaching effects, one which has been implicated in other coral species to be upregulated in bleaching. But what they really found a great use for this low coverage sequencing data was building polygenic risk scores for bleaching using the top SNPs. Models that incorporated this polygenic risk score explained about 62% of the variance in visual bleaching scores in the coral reefs. So with that, take any questions.